Nucleic acid are large biomolecules that are crucial in all cells and viruses. They are composed of nucleotide, which are the monomers components, a 5-carbon sugar, a phosphate group, and a nitrogen base. The two main classes of nucleic acid are DNA and RNA. If the sugar is ribose, the polymer is RNA. If the sugar is deoxyribose, a variant of ribose, the polymer is DNA. Nitrogenous bases are molecules that act as the building blocks of genetic information in DNA and RNA. Although there are many nitrogenous bases, the five most important ones include adenine, guanine, cytosine, thymine, and uracil. There are two categories of nitrogenous bases that serve as essential components of nucleotides. These include pyrimidine and purine. The structure of pyrimidine is constituted by six-member ring made up of carbon-nitrogen atoms. This ring is constituted by carbon atoms and two nitrogen atoms in its structure, while purine is constituted by double ring system. The pyrimidine ring is a six-member carbon-nitrogen ring with alternating atom of carbon and nitrogen, while the imidazole ring contains three carbon atoms and two nitrogen atoms, forming a five-member ring structure. The structure of nitrogenous bases. The five important nitrogenous bases include adenine, guanine, cytosine, thymine found in DNA, and uracil found in RNA. It is important to note that DNA consists of adenine, guanine, cytosine, and thymine, whereas RNA consists of adenine, guanine, cytosine, and uracil. Each base binds to a specific complementary base via hydrogen bonding. Adenine binds to thymine in DNA, while in RNA, adenine binds to uracil. Adenine, being a purine base, has a double ring structure. Adenine forms complementary bonds with thymine in DNA with uracil in RNA via two hydrogen bonds. Guanine is also a purine characterized by a double ring structure. Guanine pairs with cytosine via three hydrogen bond in both DNA and RNA. Cytosine is a pyrimidine base which is formed of a single ring structure. Cytosine pair with guanine in both DNA and RNA. Thymine is another pyrimidine base similar to cytosine, having a single ring structure. In DNA, thymine pairs specifically with adenine via two hydrogen bonds. Uracil is pyrimidine which has a similar structure to that of thymine but is found in RNA. Thymine is swapped by uracil in RNA, and it's paired with adenine via two hydrogen bonds during the process of transcription.
Welcome, DNA detectives, to a molecular showdown between two tiny but mighty molecules, nucleosides and nucleotides. Get ready to unravel the mystery of their structures and functions in the genetic world. Meet the suave nucleoside, sporting a stylish combination of a nitrogenous base and a sugar molecule. It's it's like the cool kid on the block, setting the stage for the DNA drama about to unfold. Nucleosides, adenosine, the smooth operator of the nucleoside group, struts in with its adenine base and ribose sugar, ready to mingle and make connections in the cellular party. Nucleotides enter the nucleotide squad, led by the charismatic adenosine triphosphate, the ultimate energy powerhouse. With its adenosine base and the triphosphate pose, ATP is the BIP of the genetic nightclub. Nucleoside play matchmaker, bringing together base and sugars to form the foundation of nucleotides. Once those phosphate groups crush the party, nucleotides transform into the life of the cellular pesta, driving energy transfer and genetic information flow. In the battle of the molecules, nucleosides keep it simple and sweet, while nucleotides bring the extra flair with their phosphate entourage. It's a genetic dance of no other. So, dear DNA detectives, remember the tale of nucleotides and nucleosides, where simplicity meets complexity, and the dance of life unfolds in the intricate world of genetics. Keep exploring, keep learning, and keep growing to the rhythm of the molecular beat. That's a wrap on our DNA dance party. Stay curious, stay inspired, and keep deciding the secrets of the genetic universe. Until next time, keep shining bright like a nucleotide star.
Nucleic acid is a vital biomolecules that store and transmit genetic information. They are composed of long chains of nucleotides which consist of a sugar, phosphate group, and a nitrogenous base. The two primary types of nucleic acids are the DNA or the deoxyribonucleic acid and the RNA or the ribonucleic acid. DNA, or the deoxyribonucleic acid, is the molecule that carries the genetic instruction for life. It is the hereditary material in humans and almost all other organisms. The structure of DNA is the double helix. DNA is typically found as double helix consisting of two strands that run in opposite directions, and it is called the antiparallel. Nucleotides. Each nucleotide in DNA is composed of a deoxyribose sugar, a phosphate group, and one of four nitrogenous bases: the adenine or the A, the thymine or the T, the cytosine or the C, and the guanine or the G. The base pairing. Adenine pairs with thymine, and that is the A to T via two hydrogen band, and cytosine pairs with guanine, and that is the C to G via 3 hydrogen band. Replication of DNA. The genetic storage. DNA stores genetic information necessary for the development, the functioning, the growth, and also in reproduction of all living organisms and many viruses. The replication. During cell division, DNA replicates so that each new cell receives an identical copy of the genetic information. The RNA, or the ribonucleic acid, is a nucleic acid that plays a crucial role in coding, decoding, regulation, and expression of genes. Unlike the DNA, RNA is typically single-stranded and involved in various biological roles within cells. The structure of RNA is single-stranded. RNA is typically single-stranded but can form secondary structures such as hairpins and loops due to intramolecular base pairing. Nucleotides. Each nucleotide in RNA consists of ribose sugar, phosphate group, and one of four nitrogenous bases: the adenine or the A, the uracil or the U, the cytosine or the C, and the guanine or the G. Uracil replaces thymine in RNA. The types and function of RNA are the mRNA, or the messenger RNA. It carries the genetic information from DNA to the ribosomes where it serves as a template for protein synthesis. The tRNA, or the transfer RNA, transports specific amino acids to the ribosome during protein synthesis. The rRNA, or the ribosomal RNA, it combines with protein to form ribosome. And the other types are the S and RNA, or the small nuclear RNA. It involved in RNA splicing. The miRNA, or the microRNA, and the siRNA, or the small interfering RNA, involved in gene regulation. And in summary, DNA and RNA are vital nucleic acids in all living organisms. The DNA offers and transmits genetic information, while the RNA translates this information into proteins, which perform various functions in the body.
The central dogma of molecular biology is a foundation concept that explains the flow of genetic information within a biological system. Proposed by Francis Crick in 1958, the central dogma outlines the process by which the information in genes flows into proteins, DNA to RNA, RNA to proteins. The process is crucial for the expression of genetic traits and the functioning of all living organisms. The discovery of central dogma was a pivotal moment in the field of molecular biology. Before its formulation, scientists understood that genes were the carriers of hereditary information, but the mechanism by which genetic information was expressed remained unclear. Francis Crick, who discovered the structure of DNA with James Watson, proposed the central dogma to explain how genetic information flows from DNA to RNA to proteins. This concept was revolutionary as it provided a framework for understanding the molecular basis gene expression. Deoxyribonucleic acid is the molecule that contains the genetic instruction of the development, functioning, growing, and reproduction of all living organisms and many viruses. Structure of DNA, a double helix formed by nucleotide pairs, allows it to store genetic information sufficiently. The sequence of these nucleotides encodes the information necessary to construct proteins, which perform most of the function in a cell. So, transcription. It is from DNA to RNA. Transcription is the first step in the expression of genes. During transcription, a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. This process involves the following steps. 1. Initiation. RNA polymerase binds to the promoter region of the gene, signaling the start of transcription. Number 2. Elongation. RNA polymerase unzip DNA and synthesizes a complementary RNA strand from the DNA template. Number 3. Termination. The RNA polymerase reaches a termination signal and the newly formed RNA strand is released. The result of transcription in messenger or the mRNA, which carries the genetic information from the DNA in the nucleus to the ribosomes into the cytoplasm, where protein synthesis occurs.
Good day. Today, we will talk about the fascinating world of mutations, explore the groundbreaking research on nucleic acids, and unveil the potential of genetic engineering, or what we call GMOs. Mutations are the foundation of evolution. They are changes in the genetic code, the blueprint of life. This alternation can be as small as a single unit of nucleotide or as large as entire chromosomes. Mutations can occur during the cell division or be triggered by the environmental factors like radiation or chemicals. Example of mutation is the sickle cell anemia. This disease is caused by a single nucleotide mutation in the gene for beta-globin, a protein in red blood cells. This seemingly small change alters the shape of red blood cells, leading to serious health complications. And now, let's talk about the recent research on nucleic acids. Research on nucleic acids is booming. CRISPR is a powerful tool that allows scientists to edit genes with incredible accuracy. RNA therapeutics hold promise for treating genetic diseases by targeting specific gene with RNA molecules. Synthetic biology allows researchers to design and create entirely new DNA sequences, opening doors to exciting possibilities. The example on recent research on nucleic acids are the CRISPR-Cas9 and the cystic fibrosis. Cystic fibrosis is a genetic disease that affects the lung and the digestive system. CRISPR is being investigated as a potential treatment to edit the faulty gene responsible for the disease. Mutations play a role in GMO creation. Scientists can induce specific mutation or induce genes from other organisms to create GMOs with desired traits, like pest resistance or improved nutrition. While GMOs offer many benefits, ethical consideration and potential environmental impacts need careful evaluation. Mutations, nucleic acids, and GMOs are intertwined in the tapestry of life. Research continues to unlock their potential. The future holds promise for personalized medicine using gene editing, more sustainable agriculture with GMO crops, and a deeper understanding of evolution. However, responsible research and ethical considerations are crucial as we move forward. Thank you.
The central dogma of molecular biology describes the flow of genetic information within a biological system. It states that DNA transcribed into RNA, which then translated into protein. Here's an overview of the process. First is transcription. Transcription is the process by which in the genetic information in DNA is copied into a complementary RNA sequence. First step is initiation. RNA polymerase bind to specific region of DNA called promoter. The DNA strands unwind and transcription begins at the start of the site. Second is elongation. RNA polymerase moves along DNA template strand, synthesizing RNA by adding complementary RNA nucleotides. These are adenine pairs with uracil, cytosine with guanine. The RNA strands elongates in the 5 to 5 to 2 3 direction. Third is termination. RNA polymerase continues until it reaches a terminator sequence in the DNA. The RNA transcript is released and the RNA polymerase detaches from DNA. So the product is the primary transcript or pre-mRNA with may undergo processing to become mature. To become mature mRNA. The second step is translation. The translation is the process by which the sequence of mRNA molecule is used to direct the synthesis of protein. The first step of translation is initiation. The small ribosomal subunits binds to the mRNA at the start codon, which is AUG. The initiator at the RNA carrying methionine pairs with the start codon. The large ribosomal subunit attaches, forming the complete ribosome. Second step is elongation. The RNA molecules bring amino acid to ribosome, matching their anticodons with the mRNA codons. The ribosome facilitates the formation peptide bonds between amino acids, creating a polypeptide chain. Third step is termination. The process continues
until a stop codon UAA, UAG, or UGA is encountered. Release factors bind ribosomes, prompting the release of newly synthesized polypeptide chain. The ribosomal subunit is disassembled. The product is polypeptide chain that will fold into a functional protein. Transcription DNA to RNA. It occurs in the nucleus in eukaryotes. RNA polymerase synthesizes RNA from the DNA template. Translation RNA to protein. It occurs in the cytoplasm at ribosome. Ribosomes synthesize proteins using mRNA template. This flow of genetic information ensures the genetic code contained in DNA is expressed as functional proteins which perform various essential roles in the cell.